So in this chapter, we're going to talk about sex-linked traits, and we'll talk about different ways of sex determination. So for chapter 13, we're just going to focus on sections 1 and 2. So a lot of organisms are hermaphrodites, where they have the ability to produce both egg and sperm, so both male sex cells and female sex cells in the same individual. So most worms, almost all slugs and snails are hermaphrodites. Most plants are hermaphrodites, again, having both the ability to make male gametes and female gametes on the same individual, or most flowers are actually perfect flowers where you have both the male parts and the female parts in the same flower. Sex determination is any biological system that is going to determine which sexual characteristics are going to develop over the course of that organism's life. So there's a lot of sex determination mechanisms. We'll go through a few of them. So ploidy sex determination is found in ants, bees, wasps, termites. So this order, Hymenoptera, of insects. So our honeybees are a classic example of this. So the males are also sometimes called drones. They are haploid, where they have 16 chromosomes. They have one set of 16 chromosomes. The females develop from are diploid. They have two sets of 16 chromosomes, so a total of 32. In order to have a female, the queen will lay an egg and it will be fertilized by the sperm of a drone. And then that fertilized egg will develop into a female worker. The alternative, right, a queen can lay an egg that is not fertilized, and that egg will develop into a drone. All right, so we can also have environmental sex determination. Some fish and most reptiles have temperature-dependent sex determination. So our friends, the sea turtles here, under higher temperatures, the probability of those eggs developing into a female increases. For crocodiles, higher temperatures increases the probability of those eggs developing into males. And so with the effects of climate change, the gender ratio we're expecting to skew more toward male, which can have some long-term effects on the viability of populations in the future, because female reproduction is the limiting factor for population growth. All right, so another way that we can show environmental sex determination is the sex ratios. So a great example of this is the Asian sheep's head wars. And in the case that the dominant, the alpha male, is killed or leaves, the oldest and most dominant female will have a change in hormones, a change in gene expression, and she will lose the female coloring, and she'll develop this big bulge on the head and actually develop testes, and so functionally changes from female to male. This process takes about 3 or 4 months to have this total change in gene expression. And this is where I ruin everybody's childhood, so sorry. Male clownfish go the other way. So if we have a mating pair, a male and a female, if the female is killed by, like, I don't know, a barracuda attack, and she's gone, the male will actually turn into the female, again, remembering that female reproduction is the limiting factor. So if we lose the female, the males can actually change. The sex determination system that we're most used to is called chromosomal sex determination. For birds, the males have two of the same kinds of sex chromosomes. So they have two Z chromosomes, whereas females have a Z and a W. So it's the females that have the different sex chromosomes, and males have two copies of similar looking sex chromosomes. So in this case, if we look at this karyotype of this bird, we see that there's a Z chromosome and a W chromosome. And so therefore, we're going to say that this is a female because they have two different kinds of sex chromosomes. And it's when meiosis occurs, right? So the male's gametes are going to end up with one of these chromosomes. They can have a Z or a Z, right? So all sperm cells are going to have Z sex chromosomes. What alleles are present on those can vary from individual to individual. And females can give either a Z or a W. So depending on how these random gametes fuse during fertilization, right? That's going to give us our probability of having 50% probability of having a male from 50% probability of having a female. All right, so now we need some more terms to explain these differences in the kinds of chromosomes that we have present here. So an autosome is any chromosome that is not a sex chromosome. So these chromosomes that are labeled 1 through 16 in whatever bird species this is, those are all autosomes. And the 17th set here are the sex chromosomes. Those are the chromosomes that are involved in sex determination. Typically, sex chromosomes are going to show up last on our karyotypes. And usually sex chromosomes don't undergo crossing over. All of the autosomes will do crossing over, right? One of those sources of genetic variability that we saw back in meiosis. The sex chromosomes tend to not do that. Okay, so sex chromosomes in mammals, and this is true for humans as well, I kind of show the opposite pattern of birds. So females are going to have two of the same sex chromosomes, where we have an X chromosome and an X chromosome. So we say biologically female humans are going to have two X chromosomes. Biologically male mammals are going to have an X chromosome and a Y chromosome. So we can look at those karyotypes here and find that this option A, we can look down here, the sex chromosomes, we have an X and a Y. This karyotype came then from a biologically male individual. The karyotype over B over here, we see an X and an X, so therefore we're going to say that this is from a biologically female individual. Mistakes of non-disjunction that occur in meiosis, right, that give rise to trisomy 18 or trisomy 21, which is also called Down syndrome, can occur also in the sex chromosomes. And actually, these are as common as Down syndrome is. We can have various types of aneuploidy in the sex chromosomes. So, for example, Turner syndrome. This is where an individual, instead of having 46 chromosomes, is going to have just 45 because they have only one X chromosome. And because there's not a Y present, we're going to call this a biological female. So this occurs in about 1 to 2,000-ish biological females. Usually, individuals with Turner syndrome are about 8 inches shorter on average than females that have two X chromosomes. Usually there's some fertility issues with these individuals. Their ovaries tend to be underdeveloped and they might even have heart or kidney defects. Kind of the other type of aneuploidy that can happen then with the X chromosome is where you actually have, instead of having two X chromosomes, you have three. And this is more common, occurs in about 1 in 1,000 individuals. Typically, what we see, people that have triploidy X are going to have just be normal. They might be a little bit taller than average. They might have some mild learning disabilities. They might have more prone to autism or ADHD, anxiety, those kinds of things, but not, but can live normal functional. Kelflinder syndrome is where you have two X chromosomes and a Y chromosome. This is more common, occurs in about 1 in 500 biological males. Typically, we see lower testosterone levels, reduced fertility, again, increased height, more prone to ADHD, anxiety, all those fun things. Here we're going to have two Y chromosomes and an X chromosome if we have Jacobs syndrome. Again, about 1 in 1,000 biological males. Again, taller, more prone to anxiety, learning disabilities like autism. You don't have to memorize all of these things, when we talk about what makes a biological male and a biological female, it's more complicated than just like X and Y or X and X.